All right, we are going to welcome our third speaker, Dr. Michael Chen. Dr. Michael Chen brings more than 30 years of experience across technology, development, operation, partnership in the biopharmaceutical industry. He has held several senior leadership roles at the companies including Synergiva Biopharma, Favio, Applied Biotech, helping drive innovation in biologic development and manufacturing technology. He earned PhD in biochemistry from University of Georgia and completed postdoc training at Scripps Research Institute. He authored 14 scientific publications and have more than 50 patents. With his experience spanning around science, operation, and innovation, he offers a unique perspective on translating biologics research into practical process solution. Let's welcome Dr. Michael Chen.
Thanks, Chrissy. Good morning, everybody. I'm Michael Chen. I'm the CEO of BPI. The company established about 6 years ago. We are using AI technology to integrate to the bioprocessing, particularly for the biologics. So today, we're going to learn a few things. Number one, we're using a site-specific integration, CLD, to get how fast the process is, how high the titer is. Particularly, we deal with those complex proteins like bispecific antibodies. Besides that, we're going to bring a few extra values to the CLD, which is how we use AI technology to evaluate the molecules' developability and also scalability. So as you can see, this is the whole ecosystem we built about 6 years. Starting from the left bottom, we can use the AI technology to generate the antibody sequence, optimize the sequence to get the PCC sequence. So those yellow, the 4 yellow platform is all AI enabling platform. So today we're just focusing on the third one, which is AlfaCell, site-specific integration. So as you know, traditional process is random integration for the cell line development. So we are using the logic, which is the cell morphology is related to the protein physics, which is a protein expression level. So we screen over 100 millions cell image to make an algorithm. So that right now we have an algorithm can predict with cell morphology to show if the hotspot, which is a high titer or stable cells. So based on all these AI lens, we can find those hotspot in genome, which is a high titer and also stable. So these slides just show that once we use the AI algorithm, we can screen over 100 million cells based on the cell images to find those hotspots, which is with high titer and stable as well. So as you can see, we use a green protein to label those hotspots on the genome, which is one single site with multi-copies. So every time once we try to produce our GOI, every GOI genome will express in the same place, so that you can see the cell pool is homogeneous. Since it is a homogeneous cell pool, we can use only 1 or 2 96 plates to screen the molecule instead of 50 or 196 Wells plates. Plus, this site has been pre-selected as a stable, so once we integrate our GOI, those molecule is stable. We don't have to waiting for 2 or 3 months stability results to final confirm our final results. So as you can see, the whole process takes about 1.5 months after transfection to get RCB. Over the last couple of years, we make over 150 different molecules, different projects. The titer is roughly about 4 to 16 grams per liter. So the first one we are talking about is titer. So as you can see, just I mentioned that, so we have 150 different molecules the median expression level is roughly about 6 grams per liter. If you look at the table in the top, in the right top, some of them, for example, the biospecific is 8.3 grams per liter, and we work with one of the top ADC company in China. We make antibodies 10 grams per liter. Particularly if you look at 15 grams per liter at 500 liter, there's one of our VIP customer. They filed IND in China and also US. And 1 month ago, the company used this product to out-licensing to the third party with $1.4 billion. So in this case, we can show this site-specific platform is acceptable by the authority, either in China or in the US. And also the industry accept this platform. Those green parts, as you can see, it's IVD company's product. Usually, as you know, the IVD's protein is very hard expressed. Usually it's in the milligram per liter level. So if you use these molecules on our system, as you can see, all those expression levels go up to the gram per liter level. And also we test this platform with biosimilar products, PD-1, dupilumab, CTLA-4. So all these ones with a high level, 10 gram, 20 gram, with 14 days or 18 days fed batch. So people will say, well, if you use the biosimilar, how's the quality compared to the original drug? So we choose the IL-4, the dupilumab, to do the product quality analysis. So as you can see, either for the charge variation or glycan profile, all the products we make on our platform compared to the original drug is very compatible. After this high titer, and the second question normally people say, once you get this RCB, how stable it is? Should we have to wait for another 3 months to evaluate the stability, or we can use this RCB, go directly for the downstream work? So since this site, once we select this site in the early part, in the research area, the process, the site has been pre-selected as stable. So this slide just shows that we express 8 different products. And then we do the 3-month standard stability evaluation. As you can see, the titer variation is plus minus 20%. So in this case, to show that the site we select, once we produce our protein, this RCB is also stable. So even for the IND submission, you still have to do 3 months stability evaluation, but you don't have to waiting for these results to do the downstream work. So once you get RCB, basically you can use this cell line to do the next step. So speed, how fast we can get this done. Since we label this site with a green protein, so as you can see, the whole cell is a green color. So once we have a GOI do the homologous transfection, the GFP gene will be replaced by the GOI. So the cell itself become from green color become non-fluorescent, non-color cells. So after about 1 week selection and also recovery, we can make it like a 5%, sometimes make it 20% of these positive cells. Since you have a color differentiation, green and non-green, we can use the FACS with the Satina, any kind of the cell sorting equipment, to specifically pick those positive cells into the 96 well. So that you don't have to use a lot of 96 well plates to screen. You need 2 or 3 plates, it's good enough. So after 96 well, go to 24, to the 6th well, to the shake-flask, basically take about 1.5 months to get this done. So if you look at the right top, this is our case studies. The real case studies showing that the blue bar just show that when we take about 40 days, 40 days to get the RCB. Now I'll take another 2 weeks to get a fed batch. So basically 60 days, we will know the final titers. So this is a case study just showing that the one of the products we're working on take only 49 days to get the titer is about 7.5 grams per liter. So as you can see in the 96 well, not sure this, okay. In the 96 well, we select 300 cells to start with. After 2 weeks, we take the top 24 to transfer to the 24 well. Then we go all the way to the 6 well, we select the top 6. So the final titer is roughly about 5 grams to 7 grams per liter. So once we use the site-specific, a lot of people were excited that since we express the protein in the same place, so the cell pool itself is homogeneous. So a lot of people say, hey, why wouldn't we just use the cell pool? But in our case, we bring a new idea is that we don't have to use the cell pool anymore. Because if you use the cell pool, it's the same thing. Once you do the transfection, you collect all those positive cells into the well. So they usually take about 1 month to get your cell pool materials. But since our RCBs are also faster, it takes about 1.5 months to get this done. So our suggestion is we don't have necessary to use cell pool materials for your tox study, for your downstream work. Waiting for about 2 more weeks, you can get the RCB itself to get this done. The reason is since the blue bar just show is a cell pool titer, the red bar is the RCB titer. So cell pool versus RCB still have some minor variation, particularly if you look at the quality. The cell pool itself, for example, if you look at the left table, this is for CIEF, the charge variation. So cell pool itself is about 49%. If you use a cell pool to make a decision, most of people think that 49% is not a good one. So maybe they dump this project and move to another one. But if you look at it, once you get the RCB itself, you make a median optimization. You can emit it back to the normal range. Same thing for the glycan profile. So you can see those blue bar. These blue parts is all those clone is the same with the same media. But if you go to the red areas, they use a change another media or immediately all those quality back to the good one. So sometimes this cell pool itself cannot give you the exact good information. So in this case, if you use the final RCB to do the cell pool, to do the toxic materials, we'll give you the firm results. So this slide is just showing that why we don't have to use the cell pool anymore. So if you look at the right chart, this is for random one. Because if a random one, if you want to use the RCB to make cell material, it takes 6 months. You get RCB, get a stable, and then you do the toxic materials. So sometimes for the emergency case, like COVID-19, for the national emergency, people just use the random cell pool, like 3.5 months to get material to go immediately to the tox study, to the clinical trial. But if you look at the left chart, left table, the 3.5 months for cell pool versus 4 months with RCB is about 1 or 2 weeks difference. But all the results will be firm. Titer is firm, the quality is also firm, and the time is almost the same. So we suggest with this new technology, don't worry about to use the cell pool to do the work. You still have some minor variations to make sometimes make a wrong decision, and you just use RCB within the within 4 months, we can get the toxic materials. All right, besides this cell pool, and very important things, because most of the people down in the audience are working with a complex molecule, bispecific, trispecific, 2-chain, 3-chains. So with our platform, we all design a very specific vectors. So as you can see, the vector, we have 4 open reading frames. And everybody, most of the people in the industry, particularly in the CLD, knows that the position of the heavy chain or light chain will affect your titer and also affect your quality of the products. So since we have these 4 open reading frames, so we can add those, make arrangement of the heavy chain and the light chain in the different position, can get the best results. So this is the 2 case studies. The top one is a 4 chains bispecific. As you can see, with different arrangement of heavy chain and light chain, if you look at the cell pool column, with different arrangement, the titer will be changed dramatically. Some is like 3.9 gram per liter, some is only 1.8 because of the position of your heavy light chain. And for the correct pairing, it's also changed. Some is like 90%, and some is just like 50%. So these results showing that the position of the heavy light chain in the vector will affect your titer and also quality. Another take-home message is if you look at the cell pool versus the monoclonal, the cell pool pretty much can predict your final one. For example, the 3.9 gram per liter in the cell pool, your final monoclonal is about 5.9. The correct pairing is 90% versus 83 in the RCB. So with these 4 open reading frame vector, we can make best of the molecule design. The bottom one is another case studies come from customer. They do by themselves, use the random one, they choose all the different way. So titer is roughly about 1 gram per liter. And once it comes to us, we make a different, with this our vector, so we make a different arrangement. So the titer is roughly about 4 gram to 5 gram per liter. This is just a summary shown as with our platform with different bispecific. Some is 2 chains, some 3 chains, some 4 chains. So the average titer for the bispecific on our platform is roughly about 5 to 6 gram per liter. And we can guarantee the correct pairing is 80% above. So this is all the technology we are working on, faster, stable, and deal with the complex. And also for the regulatory point of view, people, we are thinking about it, we are using fluorescent protein, we use the integrase, do we have a contamination? So all the work, if you look at the left side, the number 1, the host cell is from CHOK1. We use the EC-ACCs and use the Merck's CHOK1, it all works. And also we're testing the final products with green protein and integrase, 0. No contamination in the final products. And also if you look at the track record, which just was shown that one of customer filed IND in China and the US, and in the right part is the IP protection. So we have over 60 plus patents filed, and some of them are already granted. And the FTO is cleared in China and also in the US. Okay, so this is all this platform itself, technology point of view. Speed, faster, tighter. And since we have AI platform, so we can use AI platform to evaluate molecule developability before we start CLD. Normally, we think about bispecific, sometimes aggregation, solubility, and also viscosity. For the bispecific, we got a lot of customers coming to us that they have 2 targets. A is pretty good, B is good. When they put all together, aggregation, unstable, all kind of the things happening. So this is all the different case studies showing that, for example, in the top one, they have 2 chains. One is a VHH, an antibody, one is a FAB. By itself, they are all good. They have stability, they have solubility, aggregation, viscosity, everything is fine. When they all put together, if you look at the wet lab, shows precipitation. So we do have AI algorithm showing that when these 2 together, the score, we usually call a developability score, is above 1. 1 means it have a high warning possibility. So as you can see, all those red color showing that when they separate itself, it's pretty good. When they put together, they show the precipitation, unstable, anything. So we can use AI before we start CLD development work. We can pretty much predict if these 2, A and B, put together can be done. If you look at the blue parts, All those two together, the AI shows the good score is below 1, and for the wet lab shows it's no precipitation at all. The second one is also, these are all more case studies showing that we have an AI to have about 90% accuracy to predict these biospecific molecules can be done for the CLD or not. The second part is for solubility and aggregation. So if you look at the left table, so we also use the aggregation to make a score. If it's above 1, shows it's a high warning we have a high possibility to get precipitates. So if you look at the left one, all this is a high warning. And the wet lab shows its test tube is a crowdness, HPLC is an aggregation. And the right table just showing that it's below 1. It's soluble, and the wet lab just shows it's clear and less crowdness. So this is for soluble. The third one is viscosity, because a lot of administration route right now, people are willing to use the sub-Q injection instead of IV injection. And so we choose those FDA-approved products. Some of them is FDA-approved for sub-Q injection. So if you look at the red circle, this is the wet lab show that the viscosity is much lower. It's a single-digit CP. Now our AI just showing that, which is the black circle, showing that our score is also below 1. We use 1 as also cutoff. But if you look at the bottom parts, it's approved by FDA for IV injection. Normally, it's a high viscosity. So as you can see, for the wet lab shows it's above 25, even 100 CP. Our AI just also shown that it's above 1. So these results just show that we use this AI technology, AI algorithm can predict, based on your molecule sequence, if this molecule can be done for sub-Q or is a high viscosity. And post-CLD, after you do the CLD, these next few slides just show that it's transferable, because we do provide some service for the RCB development, but we need to transfer to the CDMO for the downstream work. This slide just shows that we work with one of the top CDMO company in China. So we have 2 products. In-house, we make 14 gram per liter, another one is 4.9 gram per liter. Within 1 month, 28 days, our CDMO can reproduce these results. It's about 11 gram per liter versus 14, and 4 gram versus 4.9. So in this case, showing that once we have this RCB in-house, we developed it for the customer, we can immediately tech transfer to the CDMO. Another one is for the scalable. So most of the titer we showed is in the shake-flask. And this one just showing that once we use 5 liter to reproduce these shake-flasks production, and the quality-wise, glycan profile, charge variation, pretty much the same. All right, so this is the last slide, just showing that with this site-specific platform, speeds faster, high quality, high quantity, and high titer. All right, thank you very much.
Great talk, Dr. Chen. I would like to take questions from the audience.
Great talk. So I just question about the site-specific integration platform. I think right now it's very popular. Many companies are developing this platform. We have several speakers, also posters. So what's the advantage of your site-specific integration platform? The first question. Second question is, you use AI to predict the landing pad. You have so many landing pads, I guess. Have you compared those landing pads to get a conclusion that which landing pad is better according to the chromosomal structure or morphology or any image? So you decided to calculate so that you can predict the next one.
Okay. The first one is ours compared to the others. We haven't done this one by one, but we know our difference, the differentiation ours between the others based on the public information. Number one, we use the green protein to label the site, so that once you choose your selection, you choose your RCB, we use the color differentiation. That's why we claim it's 1.5 months faster. If you don't have this color, even if this is site specific, it's still like a random. You still need a lot of work to screen to get your positive cells. So that's the thing. So we label this with the green protein. The second question is landing pad. So in the beginning, we don't know how do we do that. So we just use the green protein as a label to do the random insertion first. So you have millions different cells landing to different sites, which we don't know which one is which. Then we can use AI to figure out based on cell morphology, see which one is high titer, which one is stable. And also we have all different kind of other screening parameters. For example, can they grow well? Can they easily to do the transfection? All kind of things. Then once we finalize this one, we just fix that. So we don't use the other way, say which landing pad is best. So we just fix this landing pad to find the best position in the genome.
Thank you.
Good morning. Thanks for the talk. Great example of how AI can really drive science forward. You mentioned the pool titers are lower than RCB in the screening process. Is that due to a lot of heterogeneity at the pool level? And if so, what would you, can you comment on where that heterogeneity comes from?
You mean the titer in the pool versus the RCB's pool?
Yes.
Yes. Normally, by theory, the pool should be exactly the same because every GOI is expressed the same place, same cell. But we do find some variations among those cells. We don't know, but we think it's because during the transfection, during the growing, some cells somehow is damaged. Start from the beginning. That's why I show things. But normally, once we select those like 24, 30, top 50 is way good enough. Top 5 is way good enough for your high titer.
And then within that variability, did you look to see if they've retained all 3 or all 4 copies?
Yeah, yes. So the site is same. If you look to the originals, everything's there, but we don't know why they just didn't go very well. Yeah.
Okay, great. Thank you.
More questions?
How many copy numbers in your single clone?
It's one site with like a max with a 4 copy number.
Oh, that's good.
Yeah.
Yeah. That's why you have a little bit high titer compared to the others. Yeah. Thank you.
I have a question here. Do you have a flexibility of using dual plasmids or maybe 3 plasmids?
We tried. So we intentionally to pick the one. Because when we do this random spread, those landing pad, you have one site, you have 2 sites with 3 sites. So we remove all 2 or 3. The reason is because you have a 2, you have to make sure every time we do the transfection, you have to make sure the 2 sites has been transfected. The probability is much lower. It's hard. So we just focus on the one site with a high titer.
Great, thank you. All right, I think this is the end of our first section in the morning, and we will come back 11:15. Thank you.